Okay, so welcome to this next video in which we are discussing the classical complement pathway. Okay, so we have discussed that the classical complement pathway is going to be activated by the fixed region of an antibody, which is either IgG or IgM. So an antibody has bound to an antigen on the surface of our microbe, and now what's going to happen is the first complement protein is going to come and bind to the fixed region of this antibody here. Okay, right. So which complement protein is the first to come and bind? Well, basically it's going to be a C1 protein. Okay, so C1 is a great big complex of proteins. Okay, so it consists of three different types of proteins known as C1q, and then also C1r and C1s. Okay, so let me show you the structure of this. So basically, C1q structure looks kind of like this. It has a sort of tail region and then a head. Okay, so you're going to put 6 of these C1q proteins together. Okay, and how am I going to draw this? So you put them almost in a ring, basically. So I'll try my best to show this nicely, but it's probably going to end up looking horrendous. Okay, so here's another one. Here's another one. I'll show 3 on one side. Okay, and then we've got another one over here. Another one here. If I try and make it symmetric, that will look nice, hopefully. Okay, and then another one here. So basically you form this complex of 6 C1q proteins and you put them together. Okay, then you also put in 2 C1Rs. Okay, so we'll just draw C1R as a little blob, and then you also put in 2 times C1Ss. Okay, so let me color code each of these. So C1R we'll have in orange, okay, and C1S we'll have in green. So basically what's going to happen is that these C1Rs and C1Ss are going to associate with the bases of the C1Qs, so over here. Okay, so here are the C1Rs in orange. Okay, so 2 associate to make the full C1 complement protein, and then we've got 2 C1Ss as well in green here. Okay, right, so this complex is going to assemble on the base of the antibody, basically, and for this what I'm going to do is I'm going to show it slightly more simply. I'm going to show it as a circle with 6 of these processes coming out, like so. 1, 2, 3, and then we've got all 6. Okay, so these are just the heads of C1q, basically. Okay, and I'll use this to denote the full C1 complement complex. Okay, I might even put C1 in the middle there. So this is this full thing here, and this is going to come and bind to the base of this antibody here. So here we have our C1 complex. Okay, right. So now, once the C1 complex is assembled on the base of the antibody, it will become active. So let's colour in this C1 complex in red. Okay, so once it's bound to the fixed region of this antibody, it's now going to become active. Now, what does it do once it's active? Well, it interacts with 2 of the other complement proteins, okay? So it's going to work on C4 and also C2. So let me show these. So here we have complement 4, so C4, and also C2. Now, both of these are going to be split apart by C1, and specifically the portion of C1 which works on them is C1s. So C1s is going to break these 2 down. So it's going to firstly break down C4. Okay, well, it's actually going to do them at the same time, but we'll talk about C4 first. It's going to break it into 2 portions. Now the big portion that we've broken this into is what's known as C4b. and the small portion is C4A. Okay, so let's colour in C4 and its two components in turquoise. So C4 is in turquoise, and here this small little fragment is C4A, and this bigger fragment is C4B. Right, now C4A just diffuses off, but C4B is going to assemble onto glycoproteins which are on the surface of the microbe. Now I'm not going to show it actually over on this microbe because there isn't space over there, so I'm going to draw another microbe out down here, but you can imagine that it's the same one, okay? In fact, maybe what I'll do is, rather than drawing another microbe, I'll put the glycoprotein here. Okay, so not in pink, because that's the colour we used for the antigen. Orange will have the glycoprotein in. Okay, so this is any old glycoprotein on the surface of a microbe, which just means some protein on the surface of the microbe which has carbohydrate groups or sugar groups attached to it. Right, so C4b is going to attach to a glycoprotein on the surface of the microbe, and I will now show this. Where shall I show it? I'll put the membrane here, and now drawn bigger, here is that glycoprotein transferred down here. Whoops, and you can't see this. Right. Okay, so this is the glycoprotein having been transferred down here and also drawn bigger. Okay, so imagine that I've just cut that piece of membrane out and transferred it down here so that you can see it. Right, so what's going to happen is that once the C1, which is now active, converts C4 into C4A and C4B, the C4B here is going to bind to this glycoprotein. Okay? Now, C1 is also going to break down this C2 protein, and again it's going to break it into two fragments, and this is where there is an absolutely hideous bit of nomenclature. This confused me no end when I was learning this, because when you go to different websites, or even just different books or different sources, they all disagree on what each fragment should be called, and they each give them the exact opposite names, so I'll explain this in a moment. So, again you break C2 into a big piece and a small piece. Okay, so we'll have C2 in vivid purple here. Okay, and it's been broken into a small piece and a big piece. Now, this is what's really confusing. The old nomenclature is to call the big piece C2A and the small piece C2B. So this is what it used to be called, the old nomenclature. But this is still incredibly pervasive. People still use this. However, around a decade ago, or in fact over a decade ago, the people who were researching into complement decided that we should label all the big fragments of complement proteins, because it's a running theme that complement proteins are split into two pieces, a small piece and a big piece. And they said we should label all the big pieces with the B labeling. So they went and said, okay, this doesn't agree with our new labeling system, so let's swap them over. Let's call the small piece C2A and call the big piece C2B. Okay, so in some sources you will see the big piece referred to as C2A and the small piece referred to as C2B, and that is the form that my lecturers used. So that was the form that I was taught. But then if you go on the internet, you'll often hear people calling the big piece C2B and the small piece C2A. Now why does this matter? Well, it's because it's the big piece which is going to bind to C4b. So C4b has already attached to this glycoprotein on the surface of the microbe, and now the big piece is going to assemble on, and the small piece is going to go off. Okay, now depending on which nomenclature you want, you will either say that C2A has bound to here, or you will say that C2B has bound here. Okay, now you understand the confusion. So if you go to different sources, you will say that you will see that people say that this is a C4b C2a complex, and then if you go to a different source, you'll find that this is a C4b C2b portion. And I was just like, what? They seemed as though they were in blatant contradiction, and in my opinion that's an awful, awful piece of confusion that's there. But you can't solve it now. Once you've got this mess, and you've got half of the world saying that the big piece is C2b and the other half saying it's C2a, how do you fix it? You can't say, okay, we'll scrap this new way and just go back to the old way, because now you've got half the world using this new way, so it's just a mess, basically. But don't let it confuse you. If you go to different sources, you'll see the big piece, i.e. the piece which is important, referred to as both C2a and C2b. Okay, now because my lecturers used the old form, I like the old form, and therefore I'm going to call this big piece C2a, but you will see it referred to as C2b all over the place. If you just type in classical complement pathway on Google, you'll find diagrams which have C2b in, and then you'll also find diagrams which have C2a in, so you'll be really confused. You'll think that everyone doesn't know what they're talking about, basically. Who's right and who's wrong, and everyone's right and everyone's wrong, and everything is relative. No, that's a totally different subject. Right. Okay, so this is C4b in turquoise. We all agree on that. And then this here, we're going to call that C2a. Okay. So we will call this complex that we now have, i.e. the complex of C4b and C2a, we will call that complex the C4b 2a complex. However, as I say, you will find people referring to this as the C4b 2b complex. Right, so what's the next move then? What does this complex now, whatever you want to call it, do? Well, basically, the next move is that it's going to act on the protein C3. So along comes the next complement protein. Okay, so this is C3. Right, and I think I'll colour C3 in bright green. We haven't used bright green much. Now, what's going to happen is that this complex, this C4b2a complex, is going to break down the C3 into two portions. Okay, again, it's going to break it down into a small portion, which we'll call C3a, and it's going to break it also down into a big portion, which we'll call C3b, and both of these portions are actually important. The A portions of C4 and what we're calling the B portion of C2, they're not important, they just diffuse away. But the A portion of C3 is actually very, very important. Okay, so we'll start with what C3b does, but we'll come back to C3a here in red. Okay, so what does C3b do? Well, basically, C3b can also stick onto glycoproteins on the surface of the microbe. So the microbe will have more than just one glycoprotein on. So we've drawn one glycoprotein which the C4b stuck onto, but of course there'll be other glycoproteins. So let's say down here we have another glycoprotein here. So we'll highlight that up in orange too. Basically, C3b can stick onto glycoproteins on the surface of the microbe. Okay, so once you've formed it, it can stick onto glycoproteins. So here it is, having stuck onto this glycoprotein here. And now what does that do? Well, basically it opsonizes the microbe. So this is a fantastic word. It's one of those sort of, if they still did keyword sheets at degree level, this would be one of those keywords. Okay, so it is what's known as an opsonin, okay, and it takes part in the process of opsonization. Okay, so basically, on phagocytes, okay, so neutrophils and macrophages, and remember a phagocyte is a cell, so let me just summarize what a phagocyte is. So a phagocyte is a cell, such as a macrophage or a neutrophil, so this is a phagocyte, okay, which is capable of eating other cells. That's what phago means. phage means eating. So it's an eating cell. It's a cell which eats lots. Okay? So, basically, what will happen is if we've got our little bacterial cell here, which I'll colour in blue, okay, what can happen is the phagocyte can engulf it. It can sort of invaginate its membranes. Let me show you the process occurring. So it will invaginate its membrane, forming a little sort of phagosome in which the little bacterial cell is sitting. And then finally that little invagination will pinch off, and what you'll get is right inside the phagocyte you'll have this vesicle known as a phagosome with the bacterial cell or other type of cell, whatever it may be, inside. Okay, so this is what's known as a phagosome, this vesicle containing the bacterium. Okay, now what will happen is then this phagosome will fuse with lysosomes which contain enzymes which will break down the bacteria within the phagosome. Okay, so basically you're going to destroy the bacterium once it's been phagocytosed. So it will be engulfed into the phagosome and then it will be destroyed. Okay, right, so that's what a phagocyte does. It eats bacteria and then destroys them, basically, digests them. Okay, and examples of phagocytes are neutrophils and macrophages. So those are the two prototypical examples of phagocytes. Now basically, if you cover a bacterium in C3b by binding the C3b to the glycoproteins on the surface of that bacterium, the effect of that is to effectively make the bacterium more tasty for the phagocyte, because the phagocyte has receptors on its surface for C3b, almost like taste receptors, and if it finds C3b on the surface of the bacterium, then it will be more likely to phagocytose it than if there is no C3b on the surface. So basically, it's kind of a sign that says, look at me, I need to be eaten, basically, to the macrophage. And it's often compared to making the bacterium more tasty, basically. That's where this word opsonization comes from. Okay, so that's one of the effects of the the classical complement pathway, that it's going to make bacteria or other microbes more tasty for phagocytes such as neutrophils and macrophages, and therefore it's going to increase their phagocytosis of this microbe. Okay, right. We'll continue this discussion where we'll discuss other things that C3b does and also what C3a does in the next video.